This presentation is going to be on glycolysis and fermentation. So you can see in this table that glycolysis is the first part of cell respiration. So this does not require oxygen. This process over here does require oxygen, but glycolysis does not. And so this can take place whether we have oxygen or not around. It's going to produce 2 ATPs. That's not a very efficient way of dealing with sugar. Sugar has the potential to make maybe up to 100 ATPs. And so we only get 2 ATPs from glycolysis, so that's not very effective. We're not even super efficient when we go through the whole process. We only end up getting about 36 ATPs doing cell respiration, which we'll get into later on. But glycolysis is very inefficient at getting the energy from sugar. This is how glycolysis works. It basically splits a glucose in half, and then we add some phosphate molecules and take some phosphate molecules off. And in the process, we make a couple NADHs, and we make some ATP. So our net gain is we have 2 pyruvates, we have 2 ATPs, and we have 2 NADHs that we make in this process. We're going to see a lot of oxidation and reduction going on in the next couple chapters, and so we're going to talk about it right now. What is oxidation? Oxidation is when we lose electrons and oftentimes hydrogens. And reduction is when we gain electrons and oftentimes hydrogens. Okay, so the easy way to remember this is oil rig. Think of OIL, oil, oxidation, loss. Rig, RIG, reduction, gain. Okay, that's an easy way to remember what's going on with oxidation and reduction. So here we can see this is part of glycolysis. We can see the NADH is gaining electrons and gaining hydrogen. Okay, and so this has been reduced. Okay, when we gain, it seems kind of silly to say reduced when you're gaining, but that's just how it works. So we're reducing the NAD to NADH. At this point, whether or not we have oxygen or not will determine how things proceed. If no oxygen is available, we will go through fermentation. And if oxygen is available, a lot of organisms can do cell respiration. So let's talk about if we have no oxygen available. If there's no oxygen available, then organisms are going to go through different types of fermentation. 2 common ones, a lot of mammals will go through lactic acid fermentation in their bodies, and yeast go through something called alcoholic fermentation, where they produce alcohol and carbon dioxide as waste products. Okay? The whole purpose of fermentation doesn't produce any energy. The whole purpose of fermentation is to oxidize the NADH. Okay? We want to get that NADH back to NAD plus so that we can keep glycolysis going. You see, NAD plus is not abundant in the cell, and it gets used up pretty quick unless you have a process for restoring it. Okay, the process down here of fermentation is designed to restore the NADH back to NAD plus. So that's the whole goal of fermentation. Okay, is to get this molecule returned to NAD plus so that it can be used in glycolysis again. These are just some examples of glycolysis taking place under different circumstances. and different types of fermentation that we see. Okay, all of these fermenting processes use different types of bacteria or different organisms. Okay, you can see here's good old yeast that's producing this, but we use different bacteria, like for making cheese, we use lactobacillus, and we use different bacteria for making Swiss cheese, and we use different bacteria for making vinegar. Different bacteria and different organisms are used to make different substances. But all of these processes, this is what's going on. NADH is being converted back into NAD. That way NAD can go back into glycolysis and we can keep making a little bit of ATP. It's not a very efficient process, but you do make a little bit of ATP. Humans, when we do fermentation, are going to produce lactic acid in our muscles. And our fermentation is pretty much what we consider a turbo. It's our turbo button. Humans can do lactic acid fermentation in their muscles when there's not enough oxygen available for short periods of time, but it really doesn't last very long. There's a lot of energy left over in the products of fermentation. If you think about lactic acid in us, lactic acid can be brought back to the liver to make more pyruvate. And think about alcohol. Alcohol is used in race cars. Okay, in dragsters, they use alcohol to fly down the track. So there's lots of energy available in alcohol as well. Fermentation is not a very good process of getting the energy out of the sugar. If you think about the waste products of cell respiration, the waste products of cell respiration are water and CO2. Those two molecules don't have a lot of energy left in them. As a matter of fact, we use both of those molecules to put out fires. So they're not very flammable. There's very little energy left in them. So cell respiration is going to be a lot more efficient at getting the energy out of sugar.